Okay, okay. Oh. All right, I'm going to do that before I kill everybody with reverb. Okay, students who are here, when we go on our first break, I'm going to go through the roster and just have you sign in. I have a little bit more material I want to cover this time than my normal class because I know some of you noticed that usually our first week class is the Intro to Linux, and this is the second week class. However, our normal instructor for that is on vacation right now and won't be back till next week. So we kind of flipped the order, but that means that I'm going to need to do a little bit more with getting your computer set up. Just a quick show of hands in the room. How many people have already connected to the Hoffman cluster? Okay, how many people have not yet connected to the Hoffman cluster? I mean, just in the past. Okay, so you never connected? Okay. What? Oh, okay, you got it before, yeah. Okay, so this is sort of a part that why am I? Let me see if I can figure out why I'm randomly in the middle of. There we go. Okay, that's better. Get myself out of the middle of my slideshow. Okay, so this isn't officially part of the class. This is just something that comes up a fair amount of times. This is a problem that comes up more and more in bioinformatics. A lot of our tools tend to be open sourced. And as a consequence of that, you have to typically download from different websites that are distributing open source software. GitHub is pretty good, but if you go to sites like SourceForge, sometimes you'll find questionable advertisements. They use those advertisements to support themselves. All right, so this is actually from the page for downloading CyberDuck. How many people already have CyberDuck? A few, okay. Those who don't have it, I'm actually gonna walk everybody through downloading it and setting it up, so don't worry about that. There we go. So you'll notice there's this start download button for downloading CyberDuck, except does not actually download CyberDuck. The real buttons are down here. This is actually an ad. And these ads are really annoying because when you hit them, they'll make you you'll download something, and it's not actually going to be CyberDuck. I don't know what it's going to be, but I guarantee you don't want it on your computer. All right, another thing that has popped up during this class before and caused trouble, people have gotten this stuff. Show of hands, how many people in the room have seen that kind of thing before? Nobody's seen like a random pop-up on the internet saying your computer has malware on it? A few people have? Okay. Yeah, this comes up, this comes up. All this sort of stuff tends to come up a fair amount. So just sort of be aware on the internet there's questionable things out there. All right, this is where I start the actual workshop. Grab a quick drink of water. All right, so people out there on the internet via WebEx, can you hear me? Okay, cool. I got at least one yes, so that means I'm going out. All right, so since it looks like my audio is good, video is not that important as long as you're getting the slides feed. All right. So I call this workshop Design to Align. This is a combination of several previous Collaboratory workshops. We combine them together to get rid of some redundancy and also to make room for a few new workshops. Because of that, you're going to see that I have some slides in here that I don't even go over. There's also a few techniques that I don't go over as well because some of the techniques are things that people don't actually do that much. Is anybody out there expecting to do your own demultiplexing of reads? Okay, so I got one that looks sort of maybe-ish. Do you use a sequencing core for your reads? Most cores do the read demultiplexing for you already. I think the only one that might not is the Broad stem cell core. Good people there. I will rag on them a little bit during this thing because they tend to return your data in a completely different format than everybody else. So I will mention them a lot because I know I have a few students using them. Students joining me out there in WebEx land, chances are you will not have to deal with the kinds of issues that people with the Broad stem cell core have to work with, just because most cores now return data in kind of a standardized format. All right, so who am I? My background is actually bench biology. So most of my life has been spent modeling diseases in mice. I worked on genetics of skeletal dysplasias, lipid metabolism. My intro to informatics and sequencing was exome analysis for rare diseases, primarily skeletal dysplasias. I did a ton of mouse modeling of human disease. I also do a fair amount now of scientific computer program design. So at some point in there, I kind of became a developer. So a couple assumptions I'm going to make about you. I'm going to assume that some of you have some experience on the command line, although in this class, I'm actually not even going to assume much of that. Who here has never worked in a command line environment before? Okay, I got one hand up. So as I mentioned, usually this is taught after Sergey's class, so a lot of times people will have that. The good news is you don't absolutely have to have command line experience for this class, but it does help a little bit. If at some point you realize that you're really shaky on the command line, or especially you find that you're working on the command line and you're really afraid to sort of play with it, come talk to me. I actually have ways of getting better with that. If you want sort of a good future dealing with sequencing data, you really need to get comfortable on the command line, and you really should try and do it quickly. In all my years of doing this, the best predictor I've had for how far a student can go with sequencing is how comfortable they get on the command line and how quickly they do it. So I'm assuming a lot of you come from more of a biological background than a computational background. Who here works primarily in a lab? Yeah, that's just about everybody. Who here is not in a lab but is still primarily biology? So you're a biology student of some kind, or a biology undergrad, PhD, or postdoc? Okay. Just not in a lab yet? Okay. And who here is primarily computer or math? Okay, so you're primarily computer science? Is that your department? Okay. Which group are you working with? Oh, okay, you just wanted to start picking up some of the sequencing? Okay, it's a really good application. I'm probably going to lean on you at different points to possibly help people who are struggling with the command line, because I'm guessing you're pretty comfortable with it. Oh, okay. Excellent. What are you transitioning from? Oh, okay. All right. I'm going to assume that some of you have some computer programming experience. So who here has done some amount of computer programming? Okay, a couple people. Who here has written computer programs in Python? Okay, excellent. So a few people. Who here has written computer programs in Perl? A few people. All right. Are you guys still working in Perl? Learn Python. Yeah, I can help you with that too. That'll be the afternoon class, but there's definitely good opportunities to learn Python. And I made the transition a few years ago, and It took a few days of work, totally worth it. Who here has written computer programs in Java? Okay, one person, they're kind of shaky. Java seems to be kind of dying off, especially in bioinformatics. Who here has written programs in C or C++? Okay, quite a few people. That language is still great for performance, although Python is doing surprisingly well in that area now too. All right, who here, and then I'm going to assume you're more interested in running your pipeline through the command line than Galaxy. So this class used to cover both command line and Galaxy. All the Galaxy material got shifted off to a Galaxy-specific class. I've used Galaxy before. Honestly, I don't like it. It's really good if you want to learn how to sequence without putting in much effort to learning the computational side. But Galaxy hides a lot of things from you. It has a tendency to sometimes automate your mistakes. And honestly, once you get comfortable in command line, Galaxy feels super inefficient. It feels like trying to play violin with a fish. All right, and the last thing I'm going to assume here is every potential next generation sequencing application is of interest to students in this class. So I don't mean that every individual student I'm going to assume is interested in every application, but I'm going to assume that if I poll the class, I'm going to find a diversity of applications. So the one that's going to probably get the most hands up. Who here is looking to get into doing some RNA-seq? Yeah, I figured. Who here is looking to do DNA sequencing to look for variants? Okay, a few people. So do you work in human genetics? What kind of diseases are you working on? Oh, okay. Oh, okay. Okay. Hallam Lab? Oh, okay. Who here is doing microbiome? Okay, I'll talk to you actually during a break, because microbiome is actually one of my big interests right now. And I've actually been working basically all from the microbiology side all the way to the sequencing side for microbiome. So definitely I'm going to talk to you during the break a little bit. Who here is looking at doing ATAC-seq? Okay, that's fine. When I made this class up, ATAC-seq didn't exist. And actually, for that matter, microbiome sequencing was barely coming into existence. Who here is doing bisulfite sequencing? Anybody? Okay, a few people. Yeah, that one used to be a lot bigger. I think everybody kind of passed that one off to core services now. Okay, so there's a big diversity of sequencing. So the point of this class is, since there's a big diversity of sequencing applications out there, I don't go too much into application-specific. I don't go into too much application-specific knowledge. I will go a little bit, just because, as everybody probably noticed when I said RNA-seq, more hands here went up than didn't. So I do make some assumptions that you're probably going to be some interest in RNA-seq, but I try and keep it generic. But what I can tell you is that for most sequencing applications, you're going to start off obviously with an experiment and hypothesis. You're going to generate a library. You're going to generate reads. And then, actually, let me. Sorry, I just realized it's supposed to be recording, so I want to get it recording before I get too much more in depth. All right, if you give me 1 second, I'm going to see about getting this recording started. So I will be right back. Oh, actually, let me see if I can just record on this computer. All right. right, I think I may need to record from Lana's computer. I'll be right back. Are you able to get the recording started? Wait, what? Oh, are you able to record from your computer? Yeah. Oh, okay. Oh, that's perfect. So you're running the WebEx recorder. I think so. It said I set it to automatically run. Oh, perfect. Okay. Yeah. That's why I can't find a record option. Okay. No problem. One step ahead. Yes, excellent. Okay, it turns out I can't get the recording started because it's already recording on her computer. So problem solved very quickly. All right, so as I mentioned, I'm assuming that just about every application is of interest to somebody in here. But pretty much for every application, you're going to get samples. You're going to generate reads. And to generate reads, you need to generate a library. So there's a whole sequencing step involved in the middle. And then once you get reads, you're going to have to get them back in a file format that's pretty standardized. Again, I make jokes about the road core. And then you're going to probably align that to some kind of a reference genome. So we're going to start off by just walking through this pipeline. And to start off, especially when this class started, sequencing equipment was a big deal. You know, it was pretty normal for a campus, if they were lucky, to have one or two Illumina or other sequencers. Now it's pretty common for a core to have 20 of them. All right, so you got to choose. You have to choose the right platform for your study. This has pretty much become a moot point, though. So Roche 454 sequencer. Anybody seen one of those around recently? Didn't think so. Yeah, that's kind of a dead platform. ABI sequencer. Anybody seen those recently? Okay, also kind of a dead platform. Illumina sequencer. Anybody seen one of those around recently? That's kind of taken over. So that's going to be another common theme here is the technology for sequencing used to be very diverse. It's very much whittling down to, I think right now, probably 3 major platforms, each of which kind of has its own niche for different purposes, and I'll get into what those mean. So again, this goes over some of the diversity out there, and single molecule real-time sequencing, which sounds really long, so everybody just calls it nanopore or PacBio. Anybody do this kind of sequencing? Anybody know the big advantage of this kind of sequencing? Yeah, long reads. That's pretty much the advantage of it. I talk a little bit about the fact that you can detect modified bases with it. I haven't seen too many people doing that, at least in my sphere recently, but everybody loves it for really long reads. So you can read just about across a bacterial ribosomal RNA sequence with this. Or you can read across an entire repeat region of a human genome and see how long it really is. So Illumina reads tend to be on the order of hundreds of bases. Nanopore reads tend to be on the order of previously thousands, but now really it's usually like tens of thousands of bases long. So super long reads. Anybody know the big disadvantage of nanopore reads? What? Accuracy. The per base accuracy of these reads is horrible. I believe any individual base call, there's about a 13% chance that the sequencer blew the call or called the wrong base there. So if you're trying to do variant calling with PacBio, you're going to have a really, really bad day. Basically, if your sequencer is calling bogus variants more than 1 in 10 bases, that's going to get really awkward. And there are ways to error model around that, but you don't want to. Yeah, PacBio is just generally not good for trying to pick up these little sequence variants. I'll talk about it later. You can try and do high depth of coverage where you just read every single base many, many times and hope that that can correct for it. But if your big emphasis is things like looking for genomic variants, PacBio is probably not the choice for you. What would be the one genomic variant type that you would want to pick up with PacBio? Deletions, or more generally, structural variations. So if you have a chunk of chromosome that's been inverted, so it's actually been cut out and flipped around, PacBio is probably the best thing to pick that up on. Or if you have a big chunk of chromosome that's been deleted, PacBio is great for that. If you have a big chunk that's been inserted, PacBio is great for trying to read through that whole thing. But if you're just looking for that one A to T change that's causing, some weird skeletal disease in your patient, PacBio is probably a really bad choice. All right, on to the next one. Ion semiconductor, also known as ion torrent. Has anybody out there dealt with this? Okay, I didn't think so. Ion torrent shows up a lot, I've noticed, in clinical applications. Research environments tend not to like ion torrent, and honestly, ion torrent tends not to like research environments. Ion torrent tends to be fairly shut off in terms of their ecosystem. So ion torrent wants you to use their aligner. They want you to use their variant calling software. They want you to use their data analysis platform, I think called Ion Reporter. And there isn't a lot of open source software that works with ion torrent data. I think GATK stopped supporting ion torrent data when GATK was on version 1.5, and GATK is now on version 4 something. So a lot of research basically abandoned the ion torrent platform. I haven't seen too many people using it. I'm actually kind of the rare exception. I started working with ion torrent data, spent probably about 6 months pulling my hair out, and then switched over to Illumina. Because ion torrent is so closed off that I couldn't even write a lot of my own tools to deal with their data. All right, next up, pyrosequencing. Not going to go into detail on this one. Pyrosequencing, I just don't see people doing that anymore. But you can see here it has its own properties. Sequencing by synthesis. That's Illumina. That's probably going to cover 90+% of people's sequencing applications. So, and since this chart has been made, this is the technology has definitely evolved. So Illumina has the market captured. Somewhat unfortunate in some ways because probably the sequencing price hasn't come down as quickly as it would have otherwise. But to their credit, Illumina has done a good job of being very rigorous for their methods and making sure that their work is reproducible. So, well, I do have questions about some of their business practices that might keep the prices a little bit higher than one would expect. It is nice that Illumina has pretty good quality control, and they are very thorough with their job. Illumina can be very expensive, especially the more high throughput pieces of equipment. And Illumina requires some of the larger amounts of DNA relative to a lot of these sequencing methods. So for example, ion torrent is really popular among clinical also because it requires a lot less DNA to be purified. Sequencing by ligation, also known as solid. I don't think I've seen anybody do that for years. So it's on here, but I'm not going to go into it in detail. And then finally, chain termination Sanger sequencing. Anybody done this? Okay, I see one person's hand, or I see a few people's hands going up. This is just the old school sequencing. So if you dropped a set of tubes in one of the Larigen buckets or in one of the MC Lab envelopes or anything like that, or just basically sent your tubes off and then the next day you get back a chart with 4 colors on it showing you different bases, you've done chain termination Sanger sequencing. So this is the black sheep on here. This is not actually high throughput sequencing. That is not next generation sequencing. That is the original sequencing. So the advantage of this method is incredibly accurate. As long as you have good clean DNA, it's very, very accurate. Your reads come out about the length or maybe a little bit longer than you typically get with an Illumina read, but it's very expensive if you want to do a lot of this. However, people generally don't do a lot of this. People generally do a little of this. So they want to verify some part of a plasmid. They want to sequence one specific part of one specific gene. So this is very good for targeted sequencing. Also because now that it's been outsourced to a lot of labs, Typically, you just send off a sample and you pay, like $5 to $8, and you get back data the next day. All right, what you get is also dependent on the specific instrument you use. So a couple different Illumina machines. There's actually more Illumina machines than this now, and most of these machines have already been upgraded. And Illumina has the new NovaSeq platform coming out, which is going to generate just apparently insane amounts of reads per run. So what you get is dependent on the machine you use. There's also a new Illumina machine coming out. I don't know if it's out yet or when it's going to come out, but it has the footprint of a PCR machine. It has a footprint and takes up about as much space as a small quantitative PCR machine, and is designed if you just need to do a little bit of sequencing in your lab. All right, computational requirements. This is where things tend to get interesting and where people sometimes underestimate their needs a lot. So one lane on an Illumina machine can generate about 15 gigs of data, although that number has actually gone up because the machines are generating more and more sequence per lane. With NovaSeq, that's going to get even bigger. During the analysis, reads tend to be uncompressed. That's not entirely true anymore. A lot of applications will now uncompress and recompress data on the fly. So you need some extra space for these things. Once the analysis is finished, you're always going to want to keep your original reads. So just like any other, just like anything else in science, you want kind of the authoritative record of what you did. And the authoritative record of your sequencing is probably going to be the FASTQ file, and I'll explain what that is in a bit, that's sent to you by your sequencing core, and it's going to be just an untouched FASTQ. But you want to keep that around so that you have a permanent record of exactly what you got. In case anybody has questions about your data, that way they know exactly where you started. You'll want a copy of the scripts and parameters you use for your analysis. Storing this is easy. Storing this is usually storing a couple of text files, essentially. It's storing some computer scripts. So this takes up a minimal amount of space, but you always want to have this saved because that's going to describe the process of how you went from your untouched original sequence file all the way out to an alignment and all the way out to your, all the way out to the figures in your paper. Journals are getting more and more strict about making your raw data available. I think, is everybody here familiar with Retraction Watch? Okay, I see a few heads nodding. I'm guessing a few people here aren't familiar with Retraction Watch. Retraction Watch is a good site to get familiar with just because you can see mistakes other people have made. And I use mistake loosely. Occasionally those mistakes were intentional. But you can see cases where somebody basically had papers retracted, occasionally multiple papers retracted, because there was either some malfeasance or some systematic error that they didn't pick up. So it's a good place to watch to avoid making those errors yourself. It's also a good place to watch because if you notice that some papers in your field are getting retracted, it might be worth looking back at some of your, it might be worth looking back and asking yourself, hey, what kind of conclusions am I basing on these papers that just got retracted? But again, there's been a lot of high-profile retractions over the last few years. So more and more journals are requiring that a lot of the raw data be made available. You'll want to keep your alignment file, and your alignment file is going to be, typically it'll first come out in SAM format, but then you can generate it into BAM format. I'll go over what those mean at the end of class, and you'll even get to see what they look like raw, or you'll get to see what a SAM format looks like raw. BAM format is by definition not human readable. So bottom line, plan on getting some hard drives. Typically, you're going to want to store your critical files, like your raw sequencing reads. You'll want to store those on 2 separate hard drives. Anybody out there using Amazon S3? Or anybody out there using Amazon Web Services at all? Okay, I see one person. So another thing that people are going to more and more is Amazon Web Services. Amazon has something called S3, where you can essentially say, I'm going to upload my critical files here, and these files are going to be stored both in Oregon and in Germany. So you can actually have them spread out. You can actually have them stored in 2 separate locations geographically, so that even if, you know, even if there's a big earthquake in Portland and you lose the files there, you actually have duplicates of the files in Germany. It's a pretty nice system. There is a little bit of a learning curve. That's another one where if that's something you want to get into, just talk to me during a break. I can give you some pointers to get started. All right, so on a bigger scale, this is one experiment right here. And as you notice, the amount of data here would fill up a MacBook Air. So one takeaway here, don't try and do bioinformatics on a MacBook Air. You laugh, I've seen somebody try it. Don't do it. It doesn't work well. Your hard drive fills up, the computer runs really slowly and starts getting really warm. All right, so your data analysis is going to be very dependent on your application. Anybody here doing microRNAs? Okay, I have one person who looks moderately interested. MicroRNAs are really cool. MicroRNAs are also really hard to prep. I've definitely seen some of the steps that go into trying to extract them from a sample, and they can be very difficult to collect. So right now, it's a little bit dark matter. We know they're there. We know they're probably important. We just don't know how to see them really well, although we're getting better at it. But if you're doing microRNAs, you're typically dealing with a few very short reads compared to a fairly short genome, because you're typically looking at just the standardized microRNA sequences. You can do that on a MacBook Air. You can probably do that on a Raspberry Pi if you know what that is. Quick show of hands, who here knows what a Raspberry Pi is? Okay, a few people. Who here thinks that I'm talking about a dessert? Okay, well, at least nobody thinks I'm talking about a dessert. Raspberry Pi is something I'm going to talk to you about later. If you find that you're struggling with command line or want to sort of really go faster further, Raspberry Pi is an absolutely great tool. Little tiny computer. You can buy it on Amazon with a complete package for something like $60, depending on the model you get. A lot of people like them because it is just a little tiny single card computer that will run a pretty decent version of Linux. But they're absolutely great if you're learning command line or programming. So on your computer, if you're messing around with the command line, or on your Hoffman account, if you're messing around with the command line, you have to always be a little bit worried you're going to make a mistake and erase something. And that's actually what keeps a lot of people from really getting comfortable on command line, is the constant fear they're going to make a mistake that messes something up horribly. The Raspberry Pi will get you around that. You get a computer, super cheap, runs Linux. You get a command line, does everything you'd expect Linux to do. And if you accidentally type in some command like rm tacrf/, which is essentially, it doesn't actually work anymore that way, but originally if you typed that in, you would delete the entire system and everything attached to it. You don't have to worry about doing that with a Raspberry Pi, because if you do it, you rewrite the operating system to the micro SD card, plug in a new one, and you're good to go in a few minutes. So especially, I know you're transitioning to computer science. That's probably going to be, that would be a very worthwhile investment for you, probably. Oh, okay. You already have one. Do you find that it helps you with getting kind of familiar in an environment where you don't have to worry about breaking things? Yeah, that's how I started too. For me, it was also a big advantage because I was learning how to code. And there's kind of an expression I use of code fearlessly. If you're on a Raspberry Pi, you can code fearlessly, because as long as you're keeping backups of the actual code, if I run some code on there and it trashes the system, or it does something I really didn't want it to do, pop in a new card. All right. So again, miRNA, it's going to be fairly minor. If you're trying to do top hat cufflinks RNA-seq analysis, and by the way, there's much better things than top hat cufflinks if this is to your pipeline. If you're still running a top hat cufflinks-based pipeline, do some literature searches for some of the newer technologies out there. So if you're running that on a mammalian transcriptome, you're going to need a little bit more of a powerful system. And if you're dealing with something like whole genome bisulfite sequencing, or whole genome sequencing in general, but especially something like whole genome bisulfite sequencing, you're probably going to run a very large remote server. All right, coverage. So anybody here heard the term coverage thrown around when talking about genomic reads? Okay. So I'm going to take a quick step back and ask a question that everybody should remember probably from elementary school. Who here remembers sampling problems? Okay, I didn't expect to see a lot of hands. Who here remembers the teacher throwing a question at you saying, if I have a jar with 5 white balls and 5 red balls in it, starting to sound familiar? And I reach in and pull one out at random, and then you can ask all kinds of probability questions off that, right? You know, what's the probability that I pull out a red ball? What's the probability that I pull out a white ball? What's the probability that I pull out a green ball? Well, if I know I don't have green balls in there and I pull out a green ball, there was an error. So you can even talk about error modeling. So sequencing is a sampling problem. With sequencing, I'm essentially reaching into the genome, and let's say I'm doing Illumina reads, and I'm doing Illumina reads that are fragment sizes of 400 bases. I'm going to randomly pull out 400 bases from the genome, and I'm just going to read it. And I'm going to read it at some known error rate, and ideally, I should be pulling out 400 bases completely at random, although I may be doing some things to make it slightly less random. If I'm doing RNA-seq, I'm going to randomly reach into the pool of the cell's RNA, and I'm going to randomly pull out 400 bases of its RNA sequence. Anybody here think that when I randomly reach in and pull out some RNA sequence, it's going to be random? So RNA-seq has a lot of biases. And I'm going to go over a lot of those later on because those tend to be big issues and a lot of people encounter them. But coverage is kind of a big deal for sequencing. It's especially a big deal if you're doing sequencing for variant calling, which is a pretty frequent application that I've seen. So when you're designing a sequencing study, and this comes up for just about every sequencing study, designing a sequencing study would be really easy if you had infinite funding and the world had infinite sequencing capacity. However, nobody has infinite funding, and the sequencing capacity right now is definitely not infinite. So we have to start making compromises. We sometimes need to not do every sample we would ideally want to do, and we sometimes need to not do as many reads on those samples as we want. If I could if I could sequence everybody in the world, and I could sequence their entire genome out to a depth of, say, 1,000, so read every base in their genome 1,000 times to make sure I got it right, and make sure I got both copies, I'd have a really good genomic picture of the world. I'd also have a lot of hard drives, but I'd have a really good genomic picture of the world. I'd also have a really insane budget. So I can't do that. Looking at this family, I actually can't even just go through this entire family and sequence all the individuals here, despite the fact that it would, so this is from a real study, despite the fact that it would have made the study a lot easier. So I know I have at least a few genetics background people in the group. Who can tell me why I can't sequence these individuals up here? What? They're dead. So pedigrees, when aligned through somebody, that means they are dead. And it typically, typically sequencing them becomes a forensic application at that point, and is usually much lower quality. Can anybody else tell me something that's notable about this pedigree? Inbreeding. Specifically, a lot of inbreeding. extensive inbreeding. This family bypasses the Lannisters and moves straight on to the Targaryens. As far as I can see, just from this pedigree, mother and father are uncle and niece. Mother's mother and father are uncle and niece. Father's mother and father are first cousins. So pretty extensive inbreeding in this family. And keep in mind that this is just the part of the pedigree that I actually have. I didn't actually get more pedigree than this. So any genetics people out there want to tell me, in these children, about half of them have a skeletal disease, half of them don't. Anybody out there want to tell me what I'm probably looking at here in terms of how this disease was inherited? Probably not X-linked. So are you guessing that because it's only in males? So in genetics, if you see an extensive inbreeding like this, you're typically thinking a recessive disease. Extensive inbreeding like this in genetics, you're thinking recessive disease until you have a reason to think otherwise. And more specifically, you're thinking that the affected individuals have 2 copies of the same exact mutation. So that kind of gives me a good hint about what I'm looking at when I'm sequencing. That also means that I can probably get pretty far just sequencing this family right here, so parents and kids. The affected individuals should have 2 copies of the variant allele. The unaffected individuals should have 1 or 0 copies. I can also kind of cheat a little bit, and I can use a little bit of cribbing, because I can kind of tell myself, whatever gene this is, since I know these children have a skeletal disease, I can probably look in genes that are known to affect the skeleton first. And I know Wei Guang is looking at this like this looks kind of familiar because, yes, I presented this in lab meeting a couple times. So variant discovery, a lot of times you want to start off by sequencing a few candidate individuals at very high depth, and a few unaffected individuals who are genetically very close to them, also at fairly high depth. This will get you a good idea of what variants you just expect to see in the family and what variants might be causative. Once you have some candidates, you can go to low throughput sequencing, which will be fairly cheap, and do targeted sequencing of those mutations to see if they kind of play out, if they are inherited as expected. So the reason I say all this, the human genome is about 3.3 billion bases. A lot of times the mutation causing a genetic disease is 1 base. It's looking for a needle in a stack of needles. Or I should say it's looking for 1 slightly bent needle in a stack of needles. So again, variant discovery, a lot of times you're going to go looking in, you're going to sort of bias yourself towards higher number of reads because you want to make sure you don't miss a variant. If you have low depth of coverage and you miss a variant, you're quite possibly going to miss your variant of interest. So again, going back to the sampling problem. If I reach into the genome and I pull out a sequence, did I pull out the allele they got from mom, or did I pull out the allele they got from dad? I see no hands going up because the answer is I don't know. That is basically a 50-50 probability, right? Does that make sense to everyone? Let's say that I sequence that region twice. So I reach in and pull out of the genome twice. What's the probability that I sequence the same allele twice? So what's the probability that I sequenced mom's allele twice? I'm hearing people say 25%. Yeah. I know it's a little too early in the morning to be doing statistics, but it helps to start getting your mind a little bit limber and thinking about things statistically early on. All right, what are the odds that I sequence dad's allele twice? The same. So the odds that I sequence the same allele twice, either mom's or dad's, would be 50%. So if I just sequence twice, the chance that I missed an entire allele, 50%. I don't like those odds, especially because you consider as you go over the entire genome, there's going to be whole chunks of the genome that I only sequenced 1 parent's allele. So I missed an allele. Those odds start getting better if I cover everything 10 times. And with exome sequencing, a lot of times you'll have coverages of 80x. So I've read every base on average 80 times. Chances there that I read only 1 parent's allele, those start getting vanishingly tiny. All right, when you're doing quantification, things will change around a little bit. So another sequencing method I didn't bring up earlier, who here is thinking about doing ChIP-seq? Okay, one person, 2 people. That one used to be super popular and it's kind of fallen away. I know a lot of people now are doing ATAC-seq or things like that. So with quantification, well, I should start off by saying, with genomic sequencing, I expect that everything's going to be present in roughly the same amounts. Yes, there's copy number variation in the human genome. That's normal and that's to be expected. But by and large in the human genome, you expect 1 copy from mom, 1 copy from dad. If you're looking at a sex chromosome and you're looking in a woman, you still expect 1 copy from mom, 1 copy from dad. If you're looking in a man, you expect 1 X chromosome, 1 Y chromosome. So an X chromosome from mom and a Y chromosome from dad. So for quantification, that gets a little bit more interesting because in quantification, you're typically expecting to get more or less of certain segments in the library. So an example would be looking at condition A and condition B here in region 1 and region 2. We see that in condition A, region 1 is very underrepresented. Or in condition A, in samples 1, 2, and 3, region 1 is underrepresented, and condition B in samples 4, 5, and 6, it's overrepresented. In region 2, that pattern reverses. So I don't know exactly what claim you'd make here, but we have some at least convincing data there might be something going on there. In reality, would I really want to make a call based on an N of 3 per group? No, you generally don't. You generally want more of an N than that. So this looks convincing because this is the only region we're looking at. Keep in mind, most of these experiments look at the entire genome. So let's say that even with all this, I have a p-value of 0.001. So 1 in 1,000 chance that this is just by random variation. How many regions are there probably in the genome? More than 1,000? So chances of finding a region reaching a p-value of 0.001 when you're looking at thousands of regions, you're pretty much assured of finding some of them just by normal variation. So that's another way I'm going to make you think about statistics a little bit too early in the morning. I apologize for that. I know a few of you are kind of groaning about it, but it's something you have to face sooner or later. All right, so in its simplest form, you'll find people talking about coverages in 2 different ways. If somebody's talking about coverage at a specific base, they mean an absolute number. How many times was that base read in the sequencing samples? If somebody's talking about coverage over an entire genome, typically that comes up with genomic variant detection. They're talking about how many times, on average, each base in your targeted region was read. So for example, when I say an exome with coverage of 80x, that means on average, the targeted bases, so exome is just targeting the exons of the genome. On average, the exons that I tried to read, each base was read 80-fold. However, there's probably no coverage or minuscule coverage outside the exons. For whole genome sequencing, I'm typically going to hit coverages of about 20x, but that's going to be across the genome. And this topic comes up a lot in bisulfite sequencing as well. So bisulfite sequencing is trying to figure out what percentage a CG or a CHD, depending on your organism, what percentage a C in the genome was methylated. So if I read that C once, what can I say about its methylation? If I read that C once, what are the possibilities for its methylation? Its possibilities are yes or no. If I read that same C twice, so I have 2 independent reads of that C, what are the possibilities for its methylation there? 0, 50%, or 100%. If I read it 10x, well, now I can say this C is methylated 0, 10, 20, 30, 40, 50, 60, 70, 80, 90, or 100% of the time. Now I start getting some resolution in there. So for bisulfite sequencing, it becomes a big deal also. And for bisulfite sequencing, you typically want a lot of coverage. If you only read things on average 5 times for bisulfite sequencing, you start having to combine regions, and it gets a little bit messier. For whole genome bisulfite sequencing, it gets messy because you have a whole genome to worry about. All right, so for coverage, how much do you actually, oh, yes? It's actually coming from all of the above. So the question for people out there on the WebEx is, where does the read count come from, essentially? So it's a function of the machine I use. For example, some machines will generate 50 million reads per flow cell, or 50 million reads per lane in a flow cell. The upcoming NovaSeq is probably going to generate a lot more than that, from what I understand. It also comes from how much DNA you put on there. There's PCR duplication that happens. I'll go over that during the library prep discussion, but sometimes you have to amplify the DNA. So one of the people who taught one of the previous courses that became this course, at one point was doing genetics on the Anopheles mosquito. Anybody here ever tried to get mosquito DNA? Can anybody guess the big problem with getting mosquito DNA? Yes, even the biggest mosquito, there's not a lot of mosquito. So even though she was sequencing literally the whole mosquito, she still got less DNA in there than even a bad human sample. So she had to do a lot of PCR and basically amplify up the genome. So she got ultimately the same number of reads as anybody else would get, but she also figured out that a lot of those reads were basically PCR duplicates, or things that were artificially over amplified. I'll go into what those mean later. So, and I think just about everybody here was talking about doing RNA-seq. Anybody know what the most abundant RNA is in just about any cell, but especially a mammalian cell? Ribosome. In just about any mammalian cell, the most frequent RNA you're going to pull out, if you just pull out bulk RNA, is going to be ribosomal RNA. Anybody interested in studying ribosomal RNA from mammalian cells? So one time I actually had one hand go up in one class. One time in one class. What? Yeah. Well, but in her case, she wasn't actually that lucky because she was actually looking for rare sequence variants in it. So she was looking for rarities among the ribosomal RNA. Yeah, it turns out nobody's interested in finding out that 70% of your mammalian cells RNA is ribosomal RNA because everybody already knew that. Here's the problem, though, is if I just do a bulk RNA sequence from a cell, what are 70% of my reads going to go to? If I just bulk pull RNA from the cell, what's going to eat up 70% of my reads? Ribosomal RNA, which we've kind of established nobody cares about. So I have a couple options at this point. I can deal with the fact that I'm going to lose 70% of my reads to ribosomal RNA. If I get rid of the ribosomal RNA, what am I then likely going to see a lot of? We'll see a lot of mRNA. Which one in particular? Well, not poly A. That's just going to be on a lot of them. What? Not necessarily tRNAs. I'm going to see a ton of beta-actin. Who here is familiar with beta-actin? Beta-actin is one of the main components of the cytoskeleton. Just about every cell is going to make a lot of it. One case where I saw something even worse than that, I looked at somebody's sequencing data in some immature red blood cells, and she had me pull up a genomic locus and didn't tell me that what I was pulling up was in the middle of beta hemoglobin. Anybody here know what a red blood cell effectively is? It's a bag of hemoglobin that people call a cell. So it was looking at one cell, and in this case, most of its RNA was actually beta globin, and trying to pull up that region in the genome to look at the reads crashed the computer. And this was a reasonably powerful computer. I just watched the memory sort of fill up and fill up and fill up, and the computer eventually slowed down and crashed. So the lesson here is you've got a lot of molecules that are going to be very rare. You have a few molecules that are very abundant. So looking at myosin in muscles, looking at actin in just about anything, looking at ribosomal RNA in just about anything, these molecules are going to eat up a big share of your reads. Now, I will say that for ribosomal RNA, we can get rid of it. There's actually a few different methods to get rid of it, and I'll go over those a little bit later. But a lot of molecules are going to be just inherently rare. So anybody here interested in transcription factors? A few people. Transcription factors will pretty much always fall in the rare category. I've never seen a situation where transcription factor was being made in really huge amounts, even if I knew that transcription factor was critical for the cell. Transcription factors tend to be rare, even if they're important. So this kind of puts us in an interesting situation with RNA studies. Let's say that 1 in a million copies of RNA in the cell is this transcription factor I'm really interested in. And let's say that I get 500,000 reads from the cell. I have about a 50-50 chance of grabbing one of those RNAs. So I have a 50-50 chance of grabbing just one. If I have a read count in the cell of 1 million, okay, my odds are getting better. But really, if I'm looking for transcription factors in a cell, I'm going to have to do a very high depth sequencing. I'm going to have to do a lot of reads in that cell just to get a decent number of read counts on my transcription factor. Does that kind of make sense to people? In other words, if I know that I have 1 million white balls and 10 black balls in the pot, I'm going to have to reach in a bunch of times before I start approaching a point where I can say confidently, based on my sampling, there's about 10 in a million black balls in the pot. Does that kind of make intuitive sense to people now? If I just reach in once, I'm going to probably tell you it's all white. If I reach in 100 times, I'm going to tell you it's all white. If I reach in 1,000 times, probably still going to tell you it's all white. So this is where things can kind of get a little bit difficult. So again, for quantification experiments, you really need to decide how deeply you want to sequence. If you're looking at something that you don't expect to be present in very high amounts, you're going to have to reach into that pot a lot more times just to have a good probability of pulling it out enough times to where you can reasonably say, I think it's present in this quantity. All right, so I'm going to go over this part. I like to call it the least you need to know. And then we're going to take a quick 10-minute break, and I'll see about getting people signed in. All right, so what's the least you need to know here? Coverage is the number of times on average that you read each base in your template. It also describes how much sequence count you're going to want, especially for RNA-seq. If you're doing sequence variant analysis, deep coverage can compensate for a lot of sins by your sequencer or your sequencing method. The reason I say that is, even with something like PacBio's horrible error rate, if I sequence everything 500, if I sequence every base 500 times, and I know that PacBio makes about a 13% error, I can kind of error model around that. Like I can say, okay, well, this thing came up as 20% of the time it came up as the wrong thing, and it came up as always the wrong thing, that 20% of the time. That's outside PacBio's error model. That's an actual sequence variant. That said, don't try and do these little sequence variants on PacBio. It doesn't usually end well. For deep, a lot of times, because money and sequencing capacity are limited, you have to make some compromises between either the number of reads you want or the number of samples you can do. You can either do a few samples with a lot of reads, or you can do a lot of samples with a few reads. And unfortunately, that's just the state of things now. As sequencing gets cheaper, as our machines get more efficient, the compromises start becoming easier because you can get more of both. Coverage can be a property of your data sometimes. So when you assume that abundances are roughly equal, for example, in genomic DNA, if I get twice as many reads in a genomic region, I'm going to guess that I probably have a sequence duplication there, or at least a copy number variation. Coverage can also be a value of your data when abundances are not assumed to be equal. So with RNA, I know that I'm going to have a lot more beta-actin and ribosomal RNA sequence than I am, for example, my favorite Fox transcription factor. So sometimes those differences are your data, especially if you're trying to do things like RNA quantification. All right, and with that, it is 10:03, at least 10:03 in LA. Good morning, or I guess good evening or night or whatever it is to my friends in Singapore. We're going to take about a 10-minute break now. If everybody can be back at 10:15, I will see you then. And please come by really quickly so that I can get you signed in up here. Obviously, that's if you're in person. So everybody feel free to go take a break now. Women's restroom is behind the elevators over there. Men's restroom is behind the elevators, one floor up, one floor down. And like I said, at some point during the break, come and see me so I can get you signed in. Oh, let me grab a pen. What's your name?
Ryan Wong. I signed up with Lana.
When did you sign up?
She got an email by the 2nd. Actually, no, since last year.
You've been signed up since last year?
I mean, last quarter. I signed up for like.
Oh, okay. Double check with her. It might have just gotten left off because you signed up really early.
Okay.
That sometimes happens.
I'll email her again.
Okay.
Did I write the name just in case?
Go check in with her, but yeah, I can probably add you after you check in with her.
Would work, right? They're at the bottom. Thank you.
Cool. Which lab are you in?
Todd French.
Oh, okay. I know Todd. I went to grad school with Todd.
Oh, is that right?
Yeah. So you're doing microbiomics?
Yeah, so we're looking at microbiome.
Oh, cool.
Experiments for surface area.
Okay. Are you still working in Thailand?
Yeah, this collaborator. I just started in November.
I got some stuff that'll help you.
Okay, I have some questions about the nanopolar.
Yep. You, I know. Okay. Cool. Which lab are you in?
Cardiology.
Which one?
It's a cardiology lab.
Oh,
Yeah.
but which cardiology lab?
Oh, Shiv Kumar. Yeah.
Okay. I think I met him once. I used to be in cardiology.
Oh, okay, cool.
I used to be in Steve Young's lab.
Oh,
Chengwei.
Which lab are you in? Oh, okay. Yeah. Were you in the meeting I was at, or I'm trying to remember? I met with your PI and a few members of your lab a few weeks ago. Yep. Tell them I said hi.
hi. This one.
Hey, how are you?
Good.
Which lab are you in again?
In Carrie's lab. Carrie and Becca.
Oh, okay.
Yeah.
What do you guys work on? Oh, cool. Nice. And you're just starting in Comp Sci. That's right. And you were in Big Summer.
Registered because I was able to get the Hoffman account yesterday.
Oh, okay. Did you put in your application?
Yeah, but it didn't.
It's still processing?
Well, I emailed the laboratory, but I don't know if they're.
Oh, but did you do the online application for the Hoffman account? Like, did you fill out the online form?
Oh,
Okay, make sure you do that. Did Lana send you a link to the video? Okay, email Lana, tell her you need a link to the video to register for a Hoffman account, and she'll, the video will walk you through it. Yep.
Jim Matsunaga,
Yep. Which lab are you in?
hockey.
Don't know that one. Which
Yeah, I'm located at the CA.
Oh, okay.
Yeah, I'm a UCLA staff. But
Ah.
I'm looking at the CA.
Okay, cool. Pretty good.
Microtubular? Or this lab?
You guys doing any microbiome now, or?
No, we've done a little RNA-seq, and hopefully we'll be doing more of it. We had someone help us before, so now I'm trying to do the monkey.
Nice. Jessica.
Good one.
Yep. Which lab are you in?
I'm in Cheyenne class.
Oh, okay. Tell her I said hi.
Okay.
I've known her since I was an undergrad.
Oh, really?
Yeah. We used to work together in Jake Luce's lab.
Abigail Blind.
Yep. And which lab are you in? Or? I know that name, but I don't know why.
Like 2 floors downstairs. Most of his lab does stuff with like nematodes.
Okay, that's where I heard the name.
Elegans,
Yeah.
But I'm working with Daphnia, a microcrustacean that
Oh.
only recently been like sequenced. So.
Cool. Always fun when you're dealing with a non-model organism. Ah.
What's that? Oh, yeah.
Oh. Okay. Which lab are you in? Oh, okay. Yeah, so, with who? Okay, yeah, I know Christoph. Yeah. Were you there when I stopped by yesterday? I'm trying to remember. I stopped by Rui Lab yesterday, so. Yeah, because I was trying to drop off some stuff that's hopefully going to be helpful for getting RNA out of adipose tissue.
Oh, yeah, she brought that stuff. Yeah.
Yeah, that was me that brought it by, because I actually, yeah, when I'm not here, I'm actually, I work for the company that makes those things, but I'm trying to sort of punch up our white adipose capacity, because, so Karen was on my PhD committee, and I also have done a lot of RNA from white adipose tissue, and I know how annoying it can be.
Yeah, well, I'm going to start on that. Hopefully, that would be helpful for me.
Mm-hmm.
Brendan Milano?
I don't think I see you on here. When did you sign up? Okay, I think some of the people who signed up earlier, I know how these are kept. So sometimes people that sign up early sometimes get missed. Stop by with Lana. I have a feeling she'll find you on the list.
Okay. And I was actually gonna not feeling too well.
Oh, okay.
But can I still, assuming I am on work, can I still come back tomorrow?
Yes, and if you're not feeling better, definitely take advantage of the WebEx. Talk to Lana. She can also give you a link to the WebEx, so you don't have to. Okay. You can actually, yeah, yeah. You probably noticed I have some fancy equipment out here. The course is actually being streamed out.
Okay, thank you.
Ah, okay. You emailed me. Cool. Which lab are you in? Oh, okay. Yeah. Cool. All right. Okay, perfect. Anybody here who has not signed in yet?
Oh, I was emailing with Lana yesterday, so I don't see my name on the list, but we've definitely been
Okay.
going back and forth.
Okay, cool.
I'm working in Jim right now.
Oh, okay.
And
So you're also working microbiome?
actually from my school,
Yes.
I'm doing the whole genome.
Okay, you're doing whole metagenome. you trying to do any whole metatranscriptome?
No.
Okay.
So I just send Jennifer a message for my
Mm-hmm.
that I should
Okay. Was Jennifer an approved PI on the Hoffman?
know, but I'm
Did you?
waiting. My supervisor is
Yes, I would say Jake is definitely an approved PI on there. Did you fill out a form online for your account? Okay. Lana should be able to send you an email message that has instructions on how to apply for the account.
she will
Yeah. Yeah. Talk to her. She can email you the link to the video. I can also talk to Jennifer and see if I can get you a link to the video. But you definitely want to apply for a Hoffman account ASAP. Yep. Are you still in the same lab?
Yeah.
Cool.
Need to get out front.
You have plans for after already? Okay. All right. Looks like we should be resuming soon. Ah. Yep. Which lab are you in? Oh, okay. Cool. So you're trying to do a genome assembly, basically? There has to be. I know a lot of people are doing their assemblies now based more with nanopore because that's much better for assembly. Yeah, I haven't looked into that particular problem too much.
You're part of the Collaboratory, that's part of the sequencing core that
Sort of. So I used to be actually part of the Collaboratory. I actually went into industry since then, but so I'm now working at Zymo. So we probably supply some of your reagents. Because I know Todd used to work a fair amount with Justin Lin. So Justin Lin is with us as well. And yeah, and actually I was the year before before they were in grad school, so we were all sort of classmates. Yeah. But actually I have some stuff I've been meaning to show Todd, because like we have a reagent for microbiome preservation without a cold chain from like if you're shipping from Thailand. It's like RNA later, but it won't mess with your samples the way that RNA later can. RNA later, a lot of people realize this, RNA later tends to lyse the gram, like the easier to lyse gram negatives. RNA later has this horrible tendency of lysing them. And since you have to get rid of the RNA later before you can actually do an extraction, you wind up basically throwing out the baby with the bathwater. So ours is also lytic. In fact, that's actually one nice thing about it. It kills pathogens, but you don't have to get rid of it. So anything that's lysed is still kept. But yeah, basically this thing will preserve like a fecal sample for a month at room temperature, and you'll have RNA integrity, which I'm guessing if you're trying to move stuff from Thailand might be useful. Yeah, this will basically snapshot it at collection. Yeah, I can get you some samples of it. Is Todd still in second floor BSRB? Oh, okay. Cool. Yeah, shoot me an email after. Yeah, shoot us an email after class, because yeah, it would be great to see Todd again. I haven't seen him for a while. All right, I think we can pick up again. I don't see any empty-looking desks that look like they weren't empty before. All right, so I think I can pick this up again. So, all right, replicates and controls. So anybody here heard people say that thanks to the age of sequencing, we're now doing hypothesis-free research? Anybody here hear that phrase, hypothesis-free research? Anybody knows what it means? Okay, so you're nodding your head. What do you think it means? Fishing expedition. Yes, and in my experience, fishing expedition has always been kind of a negative description of research for me. It's usually what I use when I was like, you really have no idea what you're looking for, do you? So yeah, when somebody says they can go hypothesis-free, that's usually when I either try and kind of point, oh, did you have a comment or are you just stretching? So I do use unbiased as well, but usually if I use unbiased, that means like demonstrable lack of bias. So for example, if you know what you're looking for and you try and set up conditions where you're more likely to see it, but probably in a not so fair way, then I would call that biased. On the other hand, you know, if I know I'm looking for a transcription factor and I just sequence to a very high depth, so I just basically, again, reach into the pot a lot more times, I'm still unbiased. But I did set up my experiment so that something that I know I want to look at, I'll be able to look at. So yeah, I get nervous. Like if I say bias, I usually mean something very specific. Like I talk about biases all the time in microbiome, just because different microbiome methods can sometimes select for or against different organisms in ways that you didn't intend. But in general, I like to say that somebody who says they're going completely hypothesis-free is probably just not being very thorough. Every time I've had somebody say they want to sequence And, some people say, well, what's your hypothesis? And they'll say, well, really my hypothesis is there's a difference between this group of cells and this group of cells. And my response is, yes, you're probably right. In fact, that's pretty much a given. If I sequence two groups of cells that are somehow different, I'm going to find differences. And then usually I can get them to say, well, I think this group of cells is going to respond to this treatment one way, and I think this other group of cells is going to respond to the treatment this other way. It's like, okay, now we're starting to develop hypotheses. Do you have any idea how their response is going to differ? And usually they have a really good idea. It's like, well, yeah, I think it's going to be like a nonspecific inflammatory response, or I think it's going to be very similar to a response to free radicals. Like, okay, so we actually do have a hypothesis. I said, do you know the members of the pathway you're looking for? Yes. Okay, we really have a hypothesis here. So, let me restart my timer real quick. So, in my book at least, nobody has convinced me that there's such a thing as hypothesis-free research, at least not in any way that has a good likelihood of achieving any of its goals. So kind of a message here is when you're planning a sequencing experiment, you still need controls, which I'm going to kind of go into in detail here. It's also good to think about things that you would expect to see no matter what. So for example, if I'm looking at a cell type that I know I'm expecting to respond to a treatment as a free radical, so something oxidative, would I expect to see a strong sort of antioxidant response in the cell? Is that kind of the expected response? So yeah, so if I treated with this stuff that I know is going to create this response, and I know there's certain transcripts that are going to go way up because the cell's responding to it, and I don't see those transcripts go up, what would be my conclusion about the experiment? Something went wrong. That's actually the right answer. In my book, at least, coming back after an experiment saying uninterpretable, inconclusive, or possibly even just there was a technical problem and I can't say anything, that's actually a valid answer in my book. And usually, if somebody gives me that, I tend to trust them a lot more down the road. It's the people who are afraid to say something didn't work that scare me. And I've seen a lot of cases where people should have said something didn't work and refused to. Has anybody out there been in that kind of situation where you see somebody showing you data and you're like, I don't think your experiment even worked the way you intended. Why are you showing me this? Yeah, I'm seeing a lot of nods out there. That's a position we've all been in. It's really awkward. At least in situations where it's like entire labs that are collaborating just jump off other people's papers or like, I think this is garbage. I don't want my name on it. I've definitely done that before. So again, don't go hypothesis free and figure out things that basically look for the boring stuff. The boring stuff like, I expect to see an antioxidant response in response to this oxidative chemical. Because even though, yes, I know it's not something you can publish, if you don't see that response to your chemical, then you have to start asking yourself questions like, did I put in the wrong chemical? Or did I get supplied the wrong chemical? Or are these not the cells I thought? Or is something even weirder happening here? And if you verify the cells are exactly the ones you think they are, and if you verify, you mass spec the chemical and the chemical is exactly what you think, and for some reason these cells are reacting in a completely unexpected way, then you just got yourself something new to study. I think there's probably more Nobel Prizes that started that way than didn't. All right, so replicates. This is the other important part. So I talked a little bit about controls. The fact that you should be looking for kind of the boring stuff that you expect all the time, and if you don't see it, then you have to start questioning everything. Replicates. You got to have replicates. So science is based off of reproducibility. And again, it's also a sampling problem. If I say that this mouse and this mouse are, if I say that my black mouse and my tan mouse are going to react differently when I put them on a high-fat diet, the response is yes, they probably will, because you know, I'm not saying what strains they are, but inbred mice all have the same coat color. A black mouse and a tan mouse are probably two different inbred strains, and you probably will be able to find a difference in how they respond to a high-fat diet. And if I just say this one black mouse and this one tan mouse, well, that's actually just two individuals. I could probably find differences even between two mice of the same strain and how they respond to a high-fat diet. So this is where we start talking about how many mice do we need in one group versus the other group to start making conclusions. Or another example I like to give, or actually two more examples I like to give. Number one is, what happens if one of your mice has a cold on the day you collect tissue? And the other one I like to say is, we did our drug study. 1/3 of the mice responded very well to the treatment. 1/3 of the mice did not have any response. And the third mouse ran away. If you ever find yourself thinking that those phrases apply to a study you're looking at, then you're probably looking at a situation of being underpowered or not having enough replicates. So without replicates, what are you going to miss? So you can miss what's called biological noise or biological variation. And that's just the fact that two mice of the same strain, which are very close to being genetically identical, are still going to be slightly different in how they react. You also miss technical noise or technical variation. That's basically due to inaccuracies in your measurement. You also miss sample size. Who here does their p-values using Microsoft Excel? I'm not seeing any hands going up, which is very surprising. Okay, who here has had to calculate a p-value for something? Okay, what have you guys been using? Okay, a lot more people using R. Nobody's using Microsoft Excel anymore? That's impressive. What are you using for p-values? Prism? Okay. So a lot of programs will let you calculate a p-value, and I think R and Prism will also let you do this. Will let you calculate a p-value with an n of 3 per group, or an n of 4 per group, or even an n of 5 per group. Have any of you done that before? Everybody's done it at least once, probably. And then at some point, somebody actually sits down with you and teaches you more statistics, and you find out that some stuff your statistics program will let you do, but that doesn't mean you should do it. So calculating a p-value with an n of 3, you can totally do it, and you should totally never do it. It's a completely invalid method. By the way, for those who haven't checked it out yet, if there's still seats open, sign up for Don Vaughn's statistics lecture. It's probably one of the most important courses in the series. He covers both modern statistical methods, as well as covering a lot of common mistakes that scientists make when they're dealing with statistics. So, little plug for one of the other classes. I've been through it. It's absolutely superb. You will learn a lot of stuff, and it may one day keep your name out of Retraction Watch. So, sample size, again, very important. So, technical replicate is sequencing the same sample twice. A biological replicate is sequencing multiple samples in that biological condition or that biological group. So, for example, having multiple patients in your treatment and multiple patients in your control group. If you ever try going to the FDA where you don't have multiple patients in your treatment or your control group, please let me know how that turns out. So here we see a couple examples. So we have two groups of mice, again, black and tan. From the black mice, we're sequencing the first one twice, so we have samples 1 and 2, and then sample 3 is from another black mouse. Here we have sequence 4 from the first tan mouse, and 5 and 6 from the second one, so we've sequenced that other mouse twice. So the great thing with sequencing, as I mentioned before, Illumina has done a lot of work to make sure that sequencing is highly reproducible. So sequencing typically has a very high rate of concordance between two experiments. That being said, it still doesn't hurt to do the occasional technical replicate. Especially if you're going between sequencing runs, or you're going between cores, or you're going between flow cells. Biology, on the other hand, is inherently noisy. There's a lot of randomness and a lot of stochastic process in biology. There's cage effects where in one cage of mice, for example, you typically have a dominant mouse that tends to eat more than the others, also tends to bully the others. You're going to see some biological noises, and biology is inherently noisy. It doesn't usually work like physics that way. So one common area that does generate technical noise is library preparation. Anybody here ever prepared your own sequencing library? Okay, so I'm seeing the microbiome person say yes, because a lot of microbiomics people still do their own libraries. A lot of people who are doing RNA don't do their own libraries. How many of you are getting your libraries prepared by a core? Okay, that's kind of what I expected. So a lot of hands go up for libraries being made by cores. Honestly, I'm a big fan of having cores do your libraries. Cores are typically very rigorous both in their quality control and in their reproducibility for library production. This gives you a couple advantages. Number one, cores will generally not make libraries for you if they think there's something wrong with your sample. So if they think the DNA is low quality, or if they think there's not enough, they're very good about making sure that they're not going to even make a library for something they think isn't going to work right or think will be bad. The other thing is cores tend to be very strict with their library preparation SOPs. So, for example, if I have a grad student in my lab preparing a library, and then I have another grad student preparing a library, and both those grad students over their entire careers are going to just do about 20 library preps total, there's probably going to be some technical noise introduced by the fact that neither of them do a lot of library prep, and worse than that, both of them might have some slightly different habits when it comes to library prep. Cores, on the other hand, tend to have people who just do hundreds of library preps, usually hundreds of library preps a year. And a lot of times they have a big emphasis on making sure that even if two library preps are done by two different people, the library preparation between them is going to be consistent. So that gets rid of a big source of technical noise. So occasionally it might seem like it's totally worth it to have to save yourself a few $100s and have the library prep done in-house. But I can pretty much guarantee that you're going to have a few experiments that get tanked because of some kind of error in library prep. And it suddenly doesn't seem so cheap when you realize that that's a few $1,000s wasted on the sequencing part. So what is the essence of library prep? So library prep is going to vary by method. If you're doing microbiome, for example, especially if you're looking for 16S sequence, your library prep is probably going to involve selectively amplifying 16S sequences from bacteria. It's also going to involve lysing bacteria. It's also going to involve a lot of that. And any one or all of those steps can potentially introduce bias or mess with your final result. So just as a quick example, I know I have at least a few bacteriologists in the crowd. What's the difference between a gram-positive and a gram-negative bacteria, besides the staining? What actually causes that difference? Gram-negatives, yeah. Gram-negatives have a relatively thin peptidoglycan wall and 2 cell membranes. Gram-positives have 1 cell membrane and a really, really thick cell wall. So who here has heard of E. coli? Shows up in food, makes people sick. Who here has heard of listeria? Also shows up in food, makes people sick. You hear about E. coli a lot of times in raw foods like lettuces and beef and that kind of thing. You hear about listeria in frozen food. So I think there was over the summer a listeria outbreak in the UK that killed like 27 people from frozen vegetables. There's actually a reason why you tend to hear about listeria in frozen food and why you tend to hear about E. coli in raw food. If you freeze E. coli, they're gram-negative, so they have a fairly thin cell wall. They pop when you freeze them a lot of times. They get ice crystals and explode. Listeria is gram-positive. It has a really thick, hard cell wall. If you freeze it, it doesn't explode. It comes back to life and makes people sick and kills some of them, unfortunately. If you're doing a microbiome preparation and you're making your microbiome library and you use a lysis method that that basically is trying to pop the bacteria by freeze-thaw, you're going to see a lot more E. coli because you explode them all, and you're going to miss a lot of the listeria because you're going to leave them whole and they're going to get flushed out. So this is something that hasn't been talked about a lot, but this is actually a big issue. Another example is some library prep methods just have trouble with sequence that is high GC content or very low GC content. You know, some library preps don't pick up sequence that is like more than 70% GC. So these are all things that people tend to run into. In addition to that, sometimes you intentionally bias your library prep. So I described before getting rid of ribosomal RNA. You're intentionally biasing your RNA library prep away from ribosomal RNA. But in that case, you know what you're doing, and that's, you know, that bias is more of a feature than a bug. Or you're making an exome, so you're pulling out specifically the expressed portions of the genome. That's a biased library prep. But in this case, again, the bias is a feature, not a bug. But in general, all library prep is going to involve creating some kind of genomic fragments that you're just going to have this big messy pool, and then ligating adapters onto these genomic fragments. So you'll generally be putting an adapter on at each end. The adapter is going to serve a few different purposes. It may or may not have, it'll generally have a molecular barcode on it, and I'll go into what that does in a bit. It's also going to have priming sites as well as binding sites. So for example, if you're doing a nanopore, basically nanopore is trying to pull a single strand of DNA through a very, very small pore in a membrane. But in order to get it started through that pore, you need something that's basically going to start pulling it through. So you need to do some library prep there. I'm actually not super familiar with nanopore library prep. I'm much more familiar with Illumina. But with Illumina library prep, the idea is still very similar. You're ligating on some pieces of DNA that are going to have a few different functions. So you have your flow cell, and you need to bind DNA to the surface of your flow cell. Your adapters are going to have these common sequences that'll bind it to the surface of your flow cell by having complementary sequences to what's on your flow cell already. You're also going to need to start, so I mentioned that Illumina is sequencing by synthesis. Synthesis, you always need a priming site in DNA. So your adapter is going to have a priming site. So generally for library prep, you start off, you get your adapters. You're going to do a size selection, at least in a lot of cases. Size selection is, let's say that I take the genome and I initially fragment up the genome. So both with RNA and DNA, you'll frequently do fragments. Anybody know good fragmenting methods? Anybody done any of their own fragmenting? Mechanical is a really good one. So mechanical usually involves a sonicator, basically hitting it with high frequency sound. Some people also like to do chemical-based methods, although because enzymes tend to have their targets, you don't get random breakage, you tend to get breakage of specific sequence. So once you've done that, you're going to do size selection, and size selection is where you then run these through a column or a gel, and you specifically pick out fragments of a certain size that have been adapterized. The reason you do this is there's some analyses that are going to make assumptions about the size of the fragment, as well as there's some downstream steps in your library prep that are going to make certain assumptions about fragment as well. You're going to do QC and quantification. So this is basically where the core or you are going to go through and make sure that your library looks like it's suitable for sequencing. The exact steps are going to depend on the method and the core. And then once you're satisfied the library looks like it's of good quality, that's when you're going to rush in and go ahead and sequence. So when you're dealing with library prep, there's a few important questions that you're going to have to answer. A single sample per lane or multiplex. So you've heard me mention multiplexing and barcodes a few times. This is where multiplexing and barcodes come into play. If I know for a fact that I need 10 million reads from a sample, but I have 50 million reads available on a flow cell, multiplexing is what can let me say, I'm going to put 5 samples on this flow cell so that I just get 10 million reads from 5 samples each. I'll go a little bit later into kind of how that works. Stranded versus unstranded. This comes up for certain applications. This is especially important, for example, if you're dealing with RNA-seq, and it's especially important if you're dealing with RNA-seq from viruses. So if I have 2 transcripts from the same genomic locus that are anti-parallel, so one transcript is made off one strand, one transcript is made off the other strand, a stranded library prep will let me know which strand generated the original read. That can be very important for attributing things to a specific transcript and say this was transcribed off of this transcript off of this strand. Humans do a little bit of anti-parallel genes, not nearly as much. Anybody here work with viruses? Okay, nobody here is working with viruses this class. Viruses are notorious for doing that. Poly-A or ribodepletion. So I mentioned before that you typically get rid of ribosomal RNAs from your sequencing studies. Poly-A and ribodepletion are the 2 methods for doing that right now. So poly-A is using poly-T to capture polyadenylated mRNAs. Ribodepletion is using targeted sequences to specifically get rid of ribosomal RNA. So what's a major disadvantage of poly-A? If you do poly-A, you're doing basically a positive selection for what? I think I hear it over there. What did you say? mRNA, yes. Poly-A pull down is going to specifically pull down mRNA. What's it not going to pull down? You're going to miss your small non-coding RNAs, you're going to miss your microRNAs, you're going to miss your large non-coding RNAs. You're going to miss quite a few things. So if you're looking for microRNAs, for example, and you do poly-A pull down to remove ribosomal RNA from your sample, you are going to come back with absolutely nothing. What's another thing that poly-A is going to select for that maybe people don't think of at first? Can anybody think of this issue? This is a less commonly thought of one, but it's actually a really big one. I've seen a lab literally tank about $20,000 on an experiment where they used poly-A and they shouldn't have, and it completely wrecked their data. So with poly-A, you're not just selecting for mRNAs, you're selecting for the 3' end of messenger RNAs. And in this case, this lab really wanted to see if there was some kind of a truncated form of their gene being made. And their gene had a transcript of over 10,000 bases. And what I saw when I looked at their data was everything. So they did poly-A pull down, and I told them, I can basically see just the 3' end of your gene, but when you're dealing with a 10,000 base mRNA, it's going to break somewhere during processing. And when it breaks somewhere during processing and you're doing a poly-A pull down, you lose everything 5' relative to the break. So what I saw when I looked at their gene and I was trying to look at the distribution of lengths, I didn't see anything that looked like it was jumping directly from the beginning to the end. I just saw this nice distribution of where the breakages were happening. This was the main thing they wanted to look at in their study, and I had to basically tell them at the end that your $20,000 massive sequencing study for the main thing you wanted to discover is uninterpretable. All right, stranded versus unstranded. I kind of went over this before. Basically, this is just determining which strand was specifically read. All right, paired end versus single end. So has anybody already been presented with the option to do paired end versus single end for sequencing? At some point, this will come up. As I mentioned before, these adapters have priming sites on them for doing sequencing by synthesis. And that's both adapters that have priming sites. So you can, in theory, sequence from both sides. And in fact, a lot of people do. So the question here is, let's say you get 50 million reads from a single lane. Is 2 times 50 million really 100 million? And the answer to that is sometimes. If you're trying to do discovery, you typically get more information. So if you're trying to do splice variants, for example, even if I don't sequence the entire length of the fragment, if I see that if my fragments are 450 bases long or whatever, if I see that paired end 1 was sequencing exon 1 and paired end 2 was sequencing exon 37, there's probably a good chance that I skip some exons in the middle there, unless I'm talking about some really short exons here. But those are calculations you can kind of make. But again, looking at it, you know, if even without sequencing what's in the middle, so I may not know exactly where the break happened, if I see exon 1 on one side and exon 37 on the other, there was probably something skipped in the middle, assuming these exons aren't tiny. If I'm looking for structural variation, so insertions or deletions, or inversions or that kind of thing, if I sequence paired end 1, and that comes from chromosome 1, and I sequence paired end 2, and that comes from chromosome 20, probably looking at here. Okay, I heard somebody say translocation. That's pretty likely translocation, especially if I keep seeing reads where paired end 1 is chromosome 1, read 2 is chromosome 20. There's a translocation there. More per base coverage. This is basically saying if I'm looking for sequence variants like genomic variants, paired end sequencing is great. I double my coverage for less than doubling my spend. You also typically get better mapping for alignment. This is usually not a good enough reason for me to actually do paired end, though. I say that because it's usually only a slight boost in mapping efficiency. Oh. On the other hand, it's not really useful if I'm quantifying. So if all I want to do is quantify the number of reads of my favorite gene, if paired end 1 maps to my favorite gene, and then paired end 2 maps to my favorite gene, what additional information did I get from paired end 2? It's not an extra read. It was paired end 1 told me that that read mapped to my favorite gene. Paired end 2 told me that that read mapped to my favorite gene. I got no additional information out of paired end 2 there. So in that case, the paired end 2 was actually useless. It was basically a waste of money and time. All right, multiplexing. So multiplexing is what I talked about before when I start saying things about barcodes and demultiplexing. So start off with your sequencing library. Only here we're going to run 3 samples in the same batch. And we notice that all of these primers here, or all these adapters, are a little bit different. Here it's a color coding. Typically on an actual adapter, it's going to be a difference of usually a base barcode. So it's usually going to be an 8 base sequence, although it might be more. But it's 8 bases, and no barcode is within 2 bases of any other barcode. Or I should say every barcode is at least 2 bases different from every other barcode. So even if you have a single base difference in the barcode, you can still positively attribute it to its origin. for multiplexing, to try and figure out how much you can multiplex, it's actually a pretty simple equation. You have a certain number of reads you get per lane or per unit in your, but generally it's going to be a lane. You have a certain number of reads you need per sample. So I know that I get, say, 50 million reads out of a lane of a flow cell, and I know that I need 10 million reads per sample. So in theory, I can multiplex this out to 5 samples in a single lane. That'll save me some money when I sequence. That being said, follow this rule of thumb. If the number of multiplexing you can do is in the dozens, knock it down by about 10%. If it's a single digit number, so for example, so for the example I gave, I said I can multiplex it 5x, I would actually multiplex it probably 4x. The reason for this is there's this law called Murphy's Law. If I'm absolutely, positively, definitely depending on a flow cell to perform at its spec of generating 50 million reads, and I really need this one flow cell to generate 50 million reads, it'll generate 40 million. It's just Murphy's Law. So to kind of guard against this, we basically will just knock it down. So if I knock it down, it will end up costing me more money, but it kind of protects me because if I do 4 reads instead of if I do 4 samples instead of 5, even if the flow cell underperforms a little bit, I'm still guaranteed pretty much to get the number of reads I want. And if I have a little bit of extra depth of coverage in the sample, at the worst, it doesn't help me at all. At the best, maybe it helps me a little bit. All right, so what's kind of the least you need to know for library prep? So biological replicates let you see the variations within the same sample group. Biology is very noisy. You're going to want a lot of biological replicates. Technical replicates let you check for variation caused by technical issues. So generally for the same, if I'm sequencing all my samples with one library prep run and one flow cell, I may not be too keen to do a technical replicate. And in this case, the sequencing is probably going to be pretty reliable. On the other hand, if I have multiple samples going in multiple days, and hold on one second. Okay. Is the WebEx feed still working? Okay, I think the WebEx feed is still working. All right, looks like I'm still feeding to WebEx, so I think that computer might have just dropped out. Sorry, I saw something on this computer that popped up that suggests the WebEx may have cut out, but I think it's still working. If not, I'll call a break in a few minutes anyway, and I'll try and deal with it. So, biological replicates, again, very important to have quite a few of those. Biology is noisy. Technical replicates, the way I like to think about technical replicates is think about a technician who makes mistakes a lot and sneezes in samples. Think about ways that they could think about places where that could happen. That's usually where you want a technical replicate. Technical replicates are basically how you detect if your technicians are making errors or if somebody's sneezing into samples or if somebody like at Illumina sneezed in the flow cell. Library prep, pretty much always going to involve ligating sequences on there. These adapters kind of contain all the sequences that you need to add onto your DNA so that it can be put in for sequencing. Library prep may also involve certain selective processes, so amplifying things, removing things like ribosomal RNAs, capturing exons. It may or may not need to be stranded. It may or may not need to be well, the reads themselves are generally paired end, but it may or may not need to be stranded depending on what you're looking for. And it may or may not need to be multiplexed. Frequently will be, yes? Not really from what I've seen. Usually the barcodes are put in by the people making library prep, and a lot of times they'll do extensive testing to make sure the barcodes aren't going to cause bizarre errors. That said, if your lab is sort of home brewing your adapters and you're home brewing your barcodes, might be worth worrying about at that point. But from the commercial kits, I wouldn't worry about it too much. All right, so I'm actually going to go out for 1 second. I just want to make sure that the WebEx feed is still running. I will be right back. Hey, is the WebEx going?
Yeah, I can hear it fine. Are people putting this in?
Oh, well, actually, the computer in that room, the WebEx just cut out.
Oh, I think it's fine.
Okay, I just want to make sure it's that computer and not the entire WebEx.
Yeah, I'll just.
Yeah, okay.
I was just cut the computer to be fine.
Yeah, it was weird. Like, I just saw the WebEx drop out on it, so.
Oh.
Okay, but yeah, mine looked like it was still on the WebEx, so I was pretty sure that was fine. Okay. Oh, okay. Cool. Well, at least most of the room's not asleep, so I'm not doing too terribly. Yeah. Although people in Singapore, I know what time it is there. I totally won't fault you if you're starting to snooze. All right, so I'm going to work through an example here, and then I think probably we can take a quick break afterwards. So let's say you're interested in comparing gene expression in a couple of knockout mice. So here we have, there we go. Here again, we have our familiar black mice and tan mice. We have this mouse here that's going to give us 2 samples, so we have a technical replicate built in. And otherwise, we have 3 samples per group. So again, looking at kind of the breakdown of our, sample counts. How many conditions? 2, black mice versus tan mice. Obviously, this may be multiplied. I could be looking in multiple tissues in each mouse group. How many biological replicates? There's 3 mice per group. Do not actually go in with 3 biological replicates. You will never get a good p-value out of that. This is at best a pilot study. Technical replicates 1. So total of 7 samples. So what's the research question? Well, that can vary a lot. If I'm looking for differential expression, that's a good case to use a single-ended library. On the other hand, if I'm looking at alternative splicing, structural variants, genomic sequence variation, that kind of thing, that's a good case to use a paired end library. So let's say that I'm doing both differential expression, which calls for single end, and alternative splicing, which calls for paired end. Which option do I choose? What? Paired end, yeah. Basically, you lose data if you go single end, but you save money. If you do paired end, you spend more money, but you gain more data. So I figure out how many reads I need per sample, and I figure out, in this case, that I need 2 lanes, so it'll probably cost me $2,000 to $4,000, although I think that price has come down a fair amount. All right, so here's one place where I'm going to talk about bias and how these samples get pooled. That tends to actually be a very difficult question and is very application dependent. Just because the number of reads I need for a microbiome sample and the number of reads I would want for, say, an exome sample, those can vary by like 2 orders of magnitude. Microbiome sample, I'm usually fine with like with like thousands and thousands of reads. An exome sample, I'm looking at millions and millions of reads. So I kind of gave the general idea of, you know, so even there, it's actually a tricky question because then I'd have to ask, you know, are you looking for alternative splicing in beta-actin, or are you looking for alternative splicing in a transcription factor that is very low abundance? And even then, I still want to ask, you know, are you expecting that your splice variant is going to be like half the population, or are you expecting your splice variant is going to be, you know, 1/1,000,000th of the population? So if you're looking, for example, for a 1 in a million splice variant in a transcription factor, I'm actually probably going to tell you don't do it, because the coverage you would need is going to be way out there. It's going to be one of those numbers with so many zeros that I don't know how to pronounce it. On the other hand, you know, if you're just trying to get an idea for splice variants in beta-actin, I'm actually going to tell you could probably run like 50 samples in a lane, because just without any enrichment or anything, most of your RNA is going to be beta-actin as long as you get rid of ribosomal. So it's a very hard question to answer as a general answer, but you kind of get the idea of what goes into the math. But trying to give a very generic answer for that is very difficult. So looking at, and for people out there in WebExLand, the question was, am I going to give specific examples of how to calculate the coverage you need? The answer I just gave was no, because there's so many variables going in there that it's really situation by situation. All right, so going back to this, there's a couple of experimental designs here, designs 1, 2, and 3. Obviously, we're talking about potential biases here. So potential design number 1, both lane 1 gets all the black mice, lane 2 gets all the tan mice. Don't do this. Can anybody see the problems inherent in this approach? So this is a case where, again, you want to ask, what happens if one of the technicians in Illumina sneezed into my flow cell? If one of my technicians sneezed into this flow cell here handling the tan mice, then the tan mice are going to be whatever biological effect I get of being a tan mouse, plus whatever biological noise I get from Illumina technician snot. So again, the approach of asking, what happens if somebody sneezed in it? Design number 2 here. I've split up my technical replicate, so that's good. So if the Illumina technician sneezed in the flow cell for design number 2, am I going to be able to detect that? Is my technical replicate going to make it really easy to detect that? Because in one of these lanes, you know, let's say they sneezed in this one. In sample 1, I'm going to see sample 1. In sample 2, it's going to be a little bit different. So at the very least, I'll be able to say, hey, there's some unexplainable differences between the technical replicates. This shouldn't exist. There's something wrong with one of these lanes. I may not be able to say which one. And I may have to come back and say, well, sample 1 and sample 2 should have been the same. They were actually very different. You know, I may not know how to quantify Illumina technician sneeze and its effect on sequencing, but I'll know that something was there. And at the very worst, I'd have to basically redo the experiment and say something went wrong here that I can't account for. I don't know why. And ideally, I'd be able to figure out at least something was wrong with the flow cell lanes. By comparison, in this experiment, I couldn't tell you if one of these lanes got sneezed in. So I'd actually just go ahead with the experiment, and I'd go ahead with the data, and I wouldn't actually know that some of the differences in these mice were because somebody sneezed in the lane. Yeah?
So at this point, you're just talking about a library that has been divided into 2 lanes, or were they technically, or were they the same sample but produced as 2 different libraries?
So a little bit of both, actually. So these are going to be multiplexed, so they're going to get that bar. So the first mouse is going to generate both of these two samples, sample 1 and sample 2. They're both going to be generated. They're going to be barcoded slightly differently. 3, 4, 5, 6, and 7 each come from one mouse. They're also going to be, at least within one lane, barcoded differently. Sample 5 here may have the same barcode as sample 1, but since they're on different lanes, it doesn't matter.
Well, I guess you wouldn't know for sure where the mistake happened. It could have happened during the preparation of the library.
Exactly. Exactly. And as I say, another possibility is the technician in the core who was doing the library prep may have sneezed in library prep number 2. That's also a possibility. I wouldn't be able to detect it there. But again, here I'd be able to detect it because I'd see some unexplainable and probably unacceptable differences between between sample 1 and sample 2, which would tell me that the same sample sequenced differently between two different preps. So that would be basically my proof that there's something was not reproducible here. Does that kind of make sense now? Okay. Design number 3 is fairly complicated. You have everything on every lane. This is more work, although this is actually the one that is kind of considered the optimal now. In fact, if you do sequencing with a core, a lot of times they'll run they'll multiplex every sample onto every lane. And there's a couple good reasons for doing this. So even if I just give a core 1 lane worth of samples, they'll still multiplex it out across all 8 lanes. This kind of covers their rear end. So imagine if I have 1 lane that just fails. Instead of losing all the data for all the samples on that lane, I've lost 1/8 of the data for all the samples in general. So some of those samples are still going to be salvageable, and there might still be some usable information. Here I can still do kind of the same thing. So here I kind of have everything technically replicated across each lane. I still have sample 1 and sample 2 as a technical replicate from 1 mouse. So if that is not reproducible, I still know something went wrong. So design 3 has another big advantage. In design 3, I can generate all these library preps at once and freeze down half of them. So I can just freeze down a chunk of this library for later. I can run lane 1 and start doing analysis on it. So let's say I run lane 1 and my biological effect was even more profound than I had anticipated. Let's say I was expecting a 10% effect. What I actually got was a 60% effect. Turns out I may not want to run lane 2 because that's way more power than I needed to answer my question. So at that point, I can save this sequencing run for later, or save it for my lab's budget for other things, or tell the PI to put it towards beer money. On the other hand, I may run lane 1 and look at the biological effect I'm interested in, and I may actually come back saying, there's not just a lack of difference here, there's a convincing lack of difference. There is such a lack of difference that I'm not going to run lane 2 because there's no way I'd ever reach anything conclusive by running more data. And probably I was wrong about my anticipated biological difference. I need to maybe save this sequencing to look at an alternative hypothesis because I was probably wrong. So you do want to be careful not to snoop on your data, like not to kind of go prematurely and say, yeah, this is bad just because with a limited sample size you didn't see a big effect. But with sequencing occasionally, you might be able to do that early if you just do one lane. Yes?
So when you're working with a core, how much will this design or input on, and do they specifically tell you if they make up their own design, will they report that back with the data, or
typically cores give you very little control over this. And actually, I'm usually okay with that. Cores tend to pick stuff that's very reproducible. And the nice thing with a core is the core has nothing invested in your results. So there's no way the core can design a layout that's going to be biasing your results one way or the other, because the core doesn't care. Yes? The cost for design 3 might be more. That being said, since everybody now goes through a core, a lot of times the core is basically decided on this, and you really don't control it. All right, so my timer is about to ding, saying that it's time to call another restroom, water, and whatever else break. So again, women's restroom is behind the elevator over there. Men's restroom is 1 floor up or 1 floor down. Mm-hmm. In terms of workshop series, kind of figure out what you want to do, and I mean, there's some workshops that are going to be general purpose, useful. Okay, so for single cell, you're doing like single cell RNA. So I don't know if anybody has a class yet on single cell. I know. So some of the workshops are generically useful. Like this one doesn't cover any specific application, but I'm covering a lot of general purpose sequencing stuff. I don't remember if I don't think we have a single cell workshop yet. Yeah, there's going to be some things that are going to be sort of generically useful. Like this class, you'll learn a lot about the sequencing technology, and you know the technology I'm describing here still gets used in single cell. There's just some additional, very complicated steps that happen first. You're using like 10x? Okay. Yeah, like 10x, you have a lot of very complicated stuff that happens first. And then some other things might be useful, like a lot of your analysis may happen in R, so taking the R class is going to be useful. You may want to someday start writing some of your own analysis tools, so the Python series may be useful. The statistics course is going to be useful for sure, just because, I mean, you need statistics knowledge no matter what you do these days.
Okay, besides
But
that, what else can I start to figure out?
RNA-seq might be interesting as well, because I'm guessing you're doing single-cell RNA.
Yeah.
So RNA-seq might be a good place, because I don't think they cover too much single-cell, but they will cover just RNA-seq in general. Mm-hmm. Yep.
Where do you get the statistics course?
Oh, that's one of the Collaboratory workshops. So the same place where you would go to sign up for this workshop. It should be called, I think, Introduction to Modern Statistics by Don Vaughn.
Oh, okay.
I don't remember its workshop number, but there should be one workshop specifically called Intro to Modern Stats. 14? Okay.
Okay.
Yeah, look for that one. I can't recommend it enough. I wish more people took it. It would prevent a lot of errors that come back to haunt people.
And then, like, I'm dealing with some SI data, and then some experiments seem to be like pair end, but then I download the data,
Mhm.
It looks like to be single. I don't know, like maybe I'm wrong.
So a lot of times, you'll see this later on, but because I'm going to give you some paired end data to play with in a bit. Paired end data, you'll get both ends as separate files.
Yeah.
So you should get one file that's going to be called, that's going to be like read one file that's going to be called read two.
Yeah, but if I download the SI file, like, are there like specific commands?
The SI file?
SI, yeah, from NCBI also.
Oh, I'm not super familiar with those, but yeah, I'd have to take a look at them. They're actually, some of the stuff I'm going to teach you is how to look at FASTQ data.
Yeah.
So you'll get an idea of what it looks like in FASTQ. You could probably find analogous information in the files you're talking about. In which case you can go back. In which case those files you should be able to figure out what's read one and what's read two in there. Yeah. Yeah, the FASTQ will tell you if you're looking at paired end one or paired end two. All right, I don't see any empty desks in here that didn't look like they were empty before, so I think we can pick it up. We have only a little bit of time left for class right now. During the break, Joanna from Zymo emailed me and reminded me of a case I had heard of in Germany. A killer known as the Phantom. So this was somebody whose DNA had turned up on all kinds of stuff at different crime scenes. I think this person's DNA turned up at like 14 different crime scenes all around, either all around Germany or all around Europe. Turning up on things like heroin syringes, cars, all kinds of stuff. The German government had like a $400,000 reward out for this person. Well, they knew it was a woman by the DNA, so they had a $400,000 reward out for this woman. It turns out the Phantom Killer was actually a female lab technician in one of the crime labs who apparently got occasionally sloppy and contaminated the stuff. So this person was turning up at random crime scenes all over the place because occasionally she made a boo-boo. Obviously, she was eventually cleared. They investigated, figured out there's no way, it tied back to the lab. It didn't tie, stuff tied back to the lab, stuff did not tie back to, and she did not tie back to the crime scenes. So fortunately, she did not get in any trouble. Well, she didn't get in any trouble with the police. She may have gotten in trouble with her supervisor. But, these kinds of accidental contaminations and people sneezing into things, it really does happen sometimes. And you always want to be somewhat aware that, people can sneeze in your samples. So I'm going to go really old school here. Anybody here ever done a silver stain protein gel? Okay, kind of the people I was expecting. I actually once did a silver stain protein gel where I figured out I could detect that somebody had sneezed near my gel. Because I was there when they sneezed, and when the gel got developed, I could actually see, yeah, it was gross. All right, so what do you get back after you do your sequencing? Well, you get back a lot of data. So again, storage needs for labs doing a lot of sequencing, not to be underestimated. So you're probably noticing at this point that the sound cut out. Unfortunately, we had a few people who did not mute their mics, and I have to re-record over this part of the section. So I'm going to just overdub these slides for you so that you don't miss the material if you're catching this on recording. So the lesson here is choose the right application for your question. And what I mean by that is, if you're looking for genetic variation, you probably want to do DNA-seq or exome. So with DNA sequencing or with exome, you're looking at just the straight DNA. You have certain expectations that come with it. You expect there to be 2 copies of every gene if you're dealing with a human, one from mama, one from papa. You expect and because of that, you're not expecting to see things present in very different amounts. compare that to, for example, RNA-seq. So with RNA-seq, you expect to see a lot of copies of certain genes and very few copies or no copies of other genes, and no copies at all of unexpressed regions. So again, DNA-seq and exome, really the best method if you're looking for genetic variation. For quantifications of different properties, you might be looking at RNA-seq for expression, miRNA if you're looking at miRNA expression levels. ChIP-seq is great for looking at the binding of proteins to DNA or chromatin states around DNA. Bisulfite-seq, that's great for looking at epigenetic states or specifically looking at DNA methylation. There are occasionally attempts being made to get a little extra out of a sequencing method. So for example, when I was a Collaboratory fellow, there were frequently requests made to take somebody's RNA-seq, and this was most frequently done with cancers. So frequently it was somebody getting RNA-seq from a tumor and asking if I can take the RNA-seq from the tumor and do a variant discovery on this. Now in theory this should work, because I mentioned exome sequencing for genetic variation. Well, you expect to find exonic sequence in RNA-seq. In reality, this method actually doesn't work nearly as well as one would think. So it encounters two major problems. The first major problem it's going to encounter is a lot of variant detection programs don't handle splice sites very well. They're designed to work on genomic DNA, and if you give them RNA-seq to analyze, sometimes they'll detect that there's a splice site there and they'll kind of know how to deal with it. A lot of times, there's a ton of bogus variant calls right around known splice sites. So that's the first problem. The second problem is a coverage problem, which I covered earlier. So coverage, again, is how many times I read something, and if I read something very few times, it's hard to call variants in it, especially knowing that even Illumina sequencing, which is generally pretty accurate calls, still has about a 2% error rate. So looking back at this, a lot of times when people would ask me to do variant calls using just RNA-seq, or I should say specifically variant discovery using RNA-seq, unfortunately I was put in the position of telling them that there's definitely no variants of interest in beta-actin. Again, beta-actin is a super highly expressed gene, guaranteed to get a lot of reads over the entire mRNA for beta-actin. And I could tell them no variants in beta-actin. They were never actually interested in beta-actin. They always said, you know, how about this transcription factor I really like? Or how about these oncogenes that I want to know if they're contributing to the tumor? Unfortunately, what I would go back to them with was telling them it's pretty much impossible to do a variant discovery on that. Now, a lot of times I could talk to them a little bit more and get them to tell me there were some very specific variants they wanted to know about. And if they wanted to know about a very specific variant, I could go back, look at the RNA, and see if I could detect that variant in the RNA being expressed. And if that variant was being expressed, I could tell them it was present. Sometimes I could tell them it looked like it was absent. And occasionally, actually pretty frequently, I had to tell them that their sequencing depth was too low, and I didn't even get enough counts in that region of the gene that I could reliably say there's a variant present or absent. So unfortunately, it doesn't work. Most of the time when a lab did this, it was because they just didn't want to spring the money for doing both exome sequencing and RNA-seq. And I get that money is tight, but if your goal is to get a certain piece of data, the best thing to do is not try and hack a sequencing platform to turn it into another sequencing platform. That never works out well. It turns out that exome sequencing exists because RNA-seq really can't do the same thing. If RNA-seq could do the same thing, then everybody would be doing it by RNA. So that is one very important lesson from this. The other time where I'd see people occasionally trying to do variant calling or variant discovery from their RNA-seq is if they had a very precious sample. So for example, dealing with an extremely small tumor or some other really tiny input piece of tissue. Like for example, I know somebody working with mosquitoes, and they didn't want to have to decide to give half their sample for DNA and half their sample for RNA. That's a scenario I can totally understand. small sample inputs are really annoying. And my answer to that is to come talk to me after class or send me an email, because the solution for this is the dual extraction, so getting both DNA and RNA very clean and basically sequencing quality DNA and RNA out of the same sample has come a pretty long way. There's definitely methods of doing this that will leave you a lot happier than trying to compromise on your sequencing method down the line. So what is the least you need to know here? Really, the least you need to know for designing an NGS experiment. First step, plan the experiment. Obviously, plan it carefully. If NGS isn't something you're experienced with, find somebody who is and see if they'll help you plan it out. Wait a little while and review the plan. And especially if you're inexperienced, review the plan with somebody who's much more experienced. If you're dealing with new territory in NGS, find somebody who's more experienced and maybe try and bribe them with a little bit of beer and see if they'll review the plan with you over a pitcher. The next step is obtain samples. Obviously, sequencing comes from somewhere, so you got to obtain your samples. Be very careful about how you obtain and handle your samples. Just as an example, I deal a lot with microbiome, especially intestinal microbiome, which is gotten from stool samples. And if you take a stool sample and immediately lyse it and process it, you see a set of microbes there that look very different than if you took that same sample, transported it at room temperature for 24 hours, and then did a lysis on it. All kinds of bacteria that are present at 24 hours weren't even really present in any noticeable amount at time 0. So microbiome is a living sample, and that happens. So be very careful about how you obtain and handle your samples. That can have a big effect. The next couple steps, making libraries and doing selections as you need, running the sequencing and doing the demultiplexing. This is pretty much always going to be done by your sequencing core. So if you're doing your own sequencing, yes, you'll be doing this yourself. But at most places, and I think UCLA, I think currently UCLA has probably about 4 or 5 different sequencing cores in various places on campus. The pipeline of making libraries, running sequencing, and doing the demultiplexing is typically handled by your core. I know that the stem cell sequencing core sometimes doesn't like to do a few of these things, but by and large, at most institutions, the cores are going to do most of these steps for you. The next step is going to be analyzing sequencing data for coverage and quality, and we're going to learn how to do this hands-on. So we'll be going over this tomorrow in class. But you always want to do quality control in your data. Make sure that your sequencing doesn't look like it's low quality. Occasionally things go wrong, and it's better if you can figure out that something's bad early instead of figuring out that it's bad after investing a lot of time and effort. Or worse than that, not noticing that it's bad and publishing with questionable data. You're going to trim and filter as needed. So that's going to be, after you analyze the quality, you might discover that you have some low quality sequences at the end. Anybody who's done Sanger sequencing before, or a lot of other sequencing methods, knows that as you get closer to the end of the read, the quality starts to fall off and occasionally become even uninterpretable. So trimming off bad quality ends will keep you from having bad quality sequence included as part of your project. And then finally, after you clean up the sequence and it's good quality and you're ready to go forward with it, that's when you're going to align it to a template. And the template is generally going to be a reference genome or some derivative of the reference genome, like consensus microRNA sequences or specifically to transcripts, that kind of thing. From there, you can pretty much go towards any application that this goes along with. So you may be doing exome or genome sequencing, you may be doing RNA-seq, maybe ChIP-seq, bisulfite-seq, or microRNA-seq. Regardless, you're going to want to align to some kind of template and then keep going with your analysis. And really, after the template alignment, the different applications start to diverge so greatly that I won't be covering those in detail for this class. So now we're going to do a quick introduction to Hoffman 2 and the Unix command line. Most of the times when I teach this class, the students have already gone through Sergey's class. So Sergey covers the intro to the Linux command line, and most of the students have some familiarity with the Linux and Unix command line. I'm aware that this quarter, because Sergey is out of town, we're doing things a little bit reversed. So in the case of this quarter, some of you may or may not have already experience with the Linux command line. So first I'm going to start off with what is Hoffman 2. Hoffman 2 is a computational cluster. That basically means you have a lot of computers that are what we call headless. So these computers don't have a keyboard, they don't have a mouse, and they don't have a screen. You interact with these computers by getting a remote shell on them and issuing commands from your own computer. You can store and process data on Hoffman 2 using the Unix command line. So Hoffman 2 is a Linux environment. Linux is great for a cluster like this because it tends to take up a lot less overhead than, for example, running a Windows machine. The amount of storage space you're going to get on the Hoffman computational cluster is going to depend pretty much on whether or not your PI or department owns their own resources on the cluster. So everyone, just by virtue of having an account, automatically gets 20 gigabytes in their home folder. They additionally get 2 terabytes in their Scratch folder. So the 2 gigabytes, or the 20 gigabytes in the home folder, that's stable. You don't have to worry about that disappearing, but it's only 20 gigs. The 2 terabytes in your Scratch folder, that's only going to be there for up to a week or two. If the files get old, so if you don't touch a file for about a week, and especially if you don't touch it for 2 weeks, that file is likely to disappear. Essentially, the Scratch folder won't let files get stale. And that's because all the users store Scratch folder space, and the way they maintain that without having to have 2 terabytes for each user, which would get terribly expensive, is only things on the Scratch folder can be stored there if they're in active use. So again, once it sits for about a week, it's potentially subject to deletion. And by about the 2-week mark, it's very likely to have disappeared. I know that I have a slide with some specifications of what you get on the Hoffman cluster, but I'm actually going to tell you that the data on the slide are obsolete. I think you get some different computing resource limitations than were made at the time of the slide. So the general structure in Linux, or even more, the general structure of how you interact with a computer. On the outside, you have what's called a GUI, or a GUI wrapper. So GUI stands for Graphical User Interface. This is how most people who have only been using computers since the 90s or 2000s, this is how they're used to interacting with computers. This is your point-and-click interface. This is the nice, pretty desktop you get. If you go down a layer, you get the shell. This is where you type in commands into your computer. So people who grew up using computers that started off in DOS mode, where you had to type in a command, this is what they were doing. Or people who now are using Linux-based computers without starting in a graphical user interface, they're starting with a shell. The reason why the shell is so nice for computational biology is you want to devote all the resources you can computationally to your computational bio, and communicating through the command line takes a lot less overhead than the computer having to constantly draw a graphical interface. Finally, at the middle of everything is the kernel. The kernel is what takes in data, what spits out data, what processes data. It's what takes in data, for example, from your keyboard, and maybe saves what you type to a file on your USB drive, or sends out what you type to a line printer. It can take in data over the network and send it to your web browser, or just about anything else. The kernel is really what is kind of air traffic control for data on your computer, and also what is doing the hard work. All right, so how does it work when you actually work on the command line? The general structure of a command line command. You start off with some command, like do the thing. That thing may be run my alignment. That thing may be write a file. It'll vary. Run a Python program. And then a lot of times you'll see, after the command do the thing, you'll have a few options. And a lot of times these options will start with a dash if they're a single letter. If they're multiple letters long, so for example here it's -F, and I'm saying with the file. So I'm giving it some kind of file to work with. or maybe some kind of file to write to. So a frequent pattern is you'll see -F if it's a single letter thing. On the other hand, if it's --, it'll frequently be multiple letters. So you could have -- and then file, the whole word written out. And then maybe you'll have some other options like -O, and then some option like do the thing with the file this way. So you enter this command into the shell. The shell interprets this command into basically machine language. The kernel gets machine language because that's what the kernel understands. And the kernel is going to say either, I know the thing they're asking, or I don't know the thing they're asking. If the kernel kind of gives you this sideways looking cat because it has no idea what you're telling it to do, it's going to spit back an error. A frequent cause of errors is just typo. So the command line tends to be both case sensitive and intolerant of typos. So if you just accidentally, if you just accidentally hit CD when you meant to hit CP, CD will change your directory. CP will copy a file. It's not going to try and guess that you're doing one thing or the other. But if you try and change directories to a file, it will throw an error at you saying that's not a directory. On the other hand, if the kernel knows the thing you're asking, so you're giving it a completely valid command, the next thing it's going to say is, does the user have privileges to do the thing? So is the thing they're trying to run, is it an executable file? If the file can be executed, then it's going to look and say, okay, they want to access this file to do it. Do they have access to the file? And if they have the appropriate kind of access to the file, then it's going to be cool. Or if you're trying to read from a file that you don't have read access to, it's going to tell you you're not allowed to. So again, if you don't have privileges to do the thing, like you're trying to read a file you don't have permission to read, or you're trying to write a file you don't have permission to write, it'll tell you there's an error. You'll get a permissions error. If you're trying to do a valid command and you have privileges on the system to do that command, then the kernel is going to execute your command. And again, hopefully it's a command you wanted to execute. Because if you accidentally told it to overwrite your data from last week that you haven't saved, you'll lose your data from last week. All right, a couple of quick Linux commands to kind of get familiar with. pwd will tell you what directory you're in. cd will change directory. The tilde is a shorthand for, the tilde is basically a shorthand for your home folder. cd ... So in Linux, ... is shorthand for the folder directly above the one I'm in. And dot is shorthand for the folder I'm currently in. Let's see. So cd ... is saying change directories to the folder, to the directory directly above this one. Look at a file, less, and then file name. The -S option will just change how it's displayed a little bit. We're not going to worry too much about that. Copy a file is CP. Delete a file is RM for remove. And you notice for all these, you're just CP, for example. CP from source to destination. Deleting a file is RM and then the file. Delete a directory is rmdir and then the directory name. Secure copy, we're not going to really use that here, but that is how you copy a file over SSH from your computer to the cluster or from the cluster to your computer. Let's see, compress a file. So that's basically using a program called gzip. There's a few different ways to do that. Uncompress a file. Basically a bunch of different commands. And what time is it? All right, so it's just about time to go. Let's see. Yeah, I'll keep going over Hoffman a little bit tomorrow at the beginning. So who here has not yet accessed the Hoffman cluster but does have their password active? Okay, so your homework is basically to install install PuTTY if you haven't, and install CyberDuck if you haven't on your computer. We'll go over tomorrow how to configure those if you don't know how. Tomorrow we're actually going to start off by accessing the cluster. For those who know how to SSH already or configure an SSH connection, your homework is to SSH into the Hoffman cluster. Don't worry too much about doing anything yet. Tomorrow, I'm going to walk everybody through getting logged in and also changing your password if you haven't already. I know the temporary passwords from the cluster tend to be hard to remember, so I can teach you how to change your password to something that you can actually remember. Anybody using a Mac or a Linux computer, you will want CyberDuck, or well, if you're using a Mac, you'll want CyberDuck. You won't need PuTTY because your computer has SSH built in. For people using Linux computers, you don't need PuTTY because your computer already has SSH. And CyberDuck doesn't work on Linux, so you're going to want FileZilla. I don't think if you're running a Linux computer, you probably already know all this already. Other than that, I'll stick around for a few minutes to answer any questions, and I will see everyone tomorrow.